Hello everyone. In this video I'll give you a full overview of all the features that we have in the Bonsai-scout app that can be used to explore interactively bonsai tree representations that we've made on high dimensional datasets. To test this for yourself, you can go to this website and then to example results, and I will now be using the cord blood cells. It's a data set which we also had quite nice results in the bonsai paper that's available for you to read. Now, what you immediately see when you open the app is that there's some information here on the data set, on the number of cells, and you see these blue buttons. You see these all over the app, for example here as well. These blue buttons give you instructions for how to use the app if something is unclear after this tutorial. On the left you see that you get information on selected nodes, and you select a node by clicking in the app. So for example, if I click here, I get the node ID, which is its cell barcode, and then the annotation that it has for the current annotation type. So in this legend, I indeed see that I have different colors for different cell types, and these were just the annotations that were provided with the original publication of this dataset. But they also made annotations based on proteins, so antibodies that they measured in these cells, and you can also look at that. And if I then click on a node, then you see that it gets a different, that you there see its annotation for that annotation type. If you want to see, well, where are all my CD4 T cells in the tree, it can sometimes be not so easy to see. So then you can use this function where you look at just the CD4 T cells, and then it will mask all the other cells and only show the CD4 T cells. So I'll just go from top left to bottom right to show you all the features. First is that you can view the trees in different ways. So most useful is if you just want to look at a subset of the tree, then you can just drag your rectangle around it and click the zoom button, and then you will just zoom on that part. A bit of an annoying thing is that we can't remove this blue rectangle by default. You have to click next to it to make it disappear. After that, you can zoom out again or zoom in again. And you can also just click the home button and go back to the start. As it says, you can scale the node size to make a tree look different a bit, and get a bit of a different view on all the individual points. But this is basically the options that are given in the viewing tab. Now this is called the layout tab because once you have a tree, it's completely determined by its topology and its branch length. But this tree you can still lay out in different ways in 2 dimensions. So in this case we use a dendrogram layout, and there you should pay attention that distances along the tree, so distances between cells, should be measured along the tree, and in the dendrogram layout only the horizontal distances matter. So it only matters how far this cell is from this cell. You have to follow the tree and you add up the horizontal distances that you travel. It's good to keep in mind, and I'll show you an example of where this is important later. I'll go back to the RNA annotations. I'm more used to that. But you can also change the layout to show it in a circular layout. And in this case we plot it on the hyperbolic disc such that it creates a fisheye effect. You can focus on part of the tree while the rest is still visible but gets squeezed in the corner. You don't have to do this hyperbolic layout if you find it confusing, but then you will have that some cells will just disappear from the figure because they're too far away, and we'll just still show them in the margin, but they're not really at this position. They're much further away, which you can see by re-zooming, and still there are some erythrocytes that are very far away from the rest. So typically I prefer the hyperbolic layout, where you can zoom, and in this case also in the viewing tab you can change the curvature to make the curvature more or less pronounced. For this data set, it's nice to look at it from the dendrogram layout. Why? Well, because normally bonsai trees are unrooted. We don't know where the root is. The likelihood of the tree is independent of where we put the root. So then a circular layout is good. However, in this data set, we have some biological knowledge that gives us a likely root. So if we zoom in on this part of the tree, we see that here's a clade of CD34 plus cells, and these are the hematopoietic stem cells. So these are in the differentiation trajectory actually the root of all the other cells. So I will now choose to put the root there, and I can do this by using the layout tweaking tab. So I can select a node that I want to make the root, and then it will redraw the root. We have to zoom out again, and now this clade of hematopoietic stem cells is at the root, and the rest comes out from there. Now something that is striking is that you would expect these cells, which are T cells, to be closer to the B cells that are over here than the myeloid cells, the monocytes that are over here. But it doesn't seem like that in the tree. This is because, as I said before, we should only measure horizontal distances. Now, this cell is just farther away from this B cell group than these T cells. But because the tree is flipped in a certain way, we can't really see that. Now, the biological conclusions we get should be independent of that, but it can be hard to get this straight in your head. That's why we offer an option in which you can flip branches to gather some intuition for which are features that are really dependent on the tree topology and which are just on how we laid it out in the 2D visualization. So here I click this node, and then I ask flip this branch, and then it's at once much more clear that these T cells are actually closer to the NK cells and the B cells than this group of monocytes. Finally, if you've created a nice visualization of your tree, you can, in the download page, download it as PNG or as SVG, click download, and you'll get that. And what we also make available is a download page where you can download the results from Sanity, which are the normalized expression values, the clustering made by cell states, which is grouping statistically indistinguishable cells together into clusters, or the results from Bonsai, which contains both the tree, a Newick file that specifies the tree topology, and posteriors for the gene expression states of every node in the tree. So going back to the tree, to Bonsai-scout, we've now had the left part, but on the right part there's more options. So we already looked at that you can change the coloring of the nodes. You can also change the node size, but in this case I didn't add numerical annotation, so I can't use that. But I can overlay the gene expression values. So for example, I have the genes that we use to reconstruct the bonsai tree on in this list, and they're ordered by Z-score, so the amount of variation they have over measurements, so the signal to noise ratio of the gene, basically. So I can select one of these genes, and then it will show you in which cells these are expressed and in which cells they're not. You can also, if you want a specific gene, sort this list by the gene ID, and then you can easily sort it using the selector on the right side. Now, for this data set, we also had available some measurements of antibodies. So because this is a site-seq data set, this is also we're also able to visualize that in the app. And for example, here there's a CD3 antibody, which is a marker of T cells, and indeed we see that it's up in the T cells, while CD11C is a marker of myeloid cells, and indeed we see that it's up there. Now, going back to the RNA annotations, what we focused on in the paper was that you have these nice reconstruction of the differentiation trajectory that is kind of known from literature, but what strikes us is that these NK cells, these purple clades, go into two groups. So one is positioned as they should be according to literature in the lymphoid part, and one seems to be in the myeloid part of the tree. Now what we did was we investigated how is this possible? What distinguishes these NK cells? So for this, the tab of marker genes is very, very useful. For example, we can first look at, well, are these NK cells actually the same? Do they have markers that convince us that these are really NK cells? Well, you can, in the marker gene tab, you can select which feature you want to look at, either at the normalized gene expression values or at the antibodies. I'll look at the normalized gene expression values now. You can select your first subset and you can select your second subset, but if you don't do that, it's by default just that the second group is all the remaining cells, and then you can click marker. So the marker gene detection is a bit of a heavy computation, so it will take a bit of time, because it is calculating for every gene the probability that if I take one cell at random from one group and the other cell from the other group, what is the probability that the gene is higher expressed in the cell from group 1 than in cell 2. So we see that these are all markers that mark the NK cells versus the rest. So indeed, I remind you that this is the group of NK cells, and these are all markers that really clearly indicate that these are NK cells and they're very different from the rest. But why then is this group of NK cells located there and this group of NK cells located there? That's an interesting question that we can also try to address using marker genes. I'll switch again to the RNA annotations, because in the marker genes you're not bound to taking subsets of the annotation. Of course, it's also possible to select subtrees. So I want to select a subtree with all the purple NK cells here, and I do that by first clicking the ancestor of this subtree and then clicking this is my ancestor. So now I've selected this node as the ancestor, but now I still have to specify on what side of this ancestor is the subtree that I'm interested in. I'm not interested in this part or in this part, I'm interested in this part. So I click any node downstream of this ancestor, click downstream, and then the app will show you by masking the other cells if you have selected the right subset. Okay, that was one subtree. Now I still want to select the other one. I zoom in on that part and let's see. I think this is the ancestor I want. I want a subtree. I click that node, I click ancestor, I click the downstream node, and indeed I have now selected these NK cells. So to check if this is true, I can just select the two groups and see that I've indeed selected two subtrees that are very highly abundant in NK cells, but are in different parts of the tree. Well, let's get the marker genes. This is a smaller group, so the marker gene calculation is much faster. And then we see that we still get very good markers, even though we confirmed that both of these groups were expressing markers for the NK cells. There are still genes that also very well separate these two groups from each other, and the other way around as well. So in the paper we conclude that these are very likely NK cells with different origins, but that you can read everything all about in our paper. Well, the final feature in the Bonsai-scout is that we can of course use the tree to cluster cells, and we let it up to you to decide how many clusters you want to create or want to look at. But let's, for example, look at 8 clusters. We then detect the groups that are maximally separated on the tree until we have 8. So specifically, we create 8 subtrees by cutting branches, so that we cut the trees in more and more pieces, and we greedily minimize the sum of pairwise distances that are still there in these subtrees. So you can do 8, you can also do 56. It's all possible, it's up to you. But once you have selected a clustering that you like, you can add it as annotation, so that it will now appear in these possible annotations which you can color the cells, and you can also now use it as marker genes. So for example, I can now ask what is actually the major difference between erythrocytes, or these cells that are very far away, which are turn out to be erythrocytes, and then I can do that by selecting its cluster, which is cluster 6, and I can compare it to, let's say, the cells at cluster 0. So now I get these two groups, I ask for the markers, and I get genes that very cleanly separate these groups from each other. So that concludes this tutorial. If you have questions, please send us an email. We're happy to receive questions, but also remarks or feature requests, and we'll get back to you. Thanks for listening, and if you want to run Bonsai on your own data, we also have a video on that, because you can use our Bonsai-scout server here to upload your own mRNA count table, and then we will run all the analyses, and you will get a link to the Bonsai-scout app where you can then explore your data.